cloud. Okay. Finally, everything is connected and working. So that's good. Okie dokie. Let's move this up here. Okay. So we've been talking in this last chapter about everything, how our hormones kind of control all the different organs and their responses. If we, kind of peek back a minute, come on, we go back, we might see like, where was it? Epinephrine versus glucagon. Epinephrine is going to stimulate the breakdown of muscle glycogen as well as liver glycogen. So you get lots and lots of sugar flooding out, including where you need it in your muscles. Whereas glucagon is just going to stimulate the liver glycogen. Muscle glycogen is constantly replenished in there and all that good stuff. Let's see here. I know, we're not going to look at that. Okay. But where I wanted to pick up, I think this is where we left off a week ago, was talking about cortisol and some other hormones in here, and just kind of look at how they affect different tissues. So cortisol acts on liver, fat, and muscle. So it is a stress hormone. If something bad is happening, like you have an infection, or you're living in fear and have constant anxiety, or if your blood glucose gets too low, so a whole bunch of different stuff in there, your body is going to release cortisol. It is going to promote fatty acid and glucose release in the body so that you have energy to deal with whatever is causing the issue, whether that's fight off an infection or something like that. Cortisol can change the types and levels of enzymes. So rather than kind of activating enzymes that are there, which is what we see with epinephrine and glucagon, cortisol is going to maybe be a longer time frame process where it's going to say, hey, make more of these enzymes. So we might expect to see that having some nuclear effects where we express more of the DNA and make more mRNA and make more of the protein in there. So for fat, it's going to increase fatty acid and glycerol release. And we may see more hormone-sensitive lipase being made or in there, or more of it being made available at least. In the liver, we're going to see Pepsi K levels rise so that we can have enhanced gluconeogenesis. And in the muscle, we're going to start breaking down proteins and export those individual amino acids to the liver where they can feed into the gluconeogenic pathway to make more glucose for energy in there. Okay. And you can see in at least two of these cases, and including glycerol, we're increasing blood sugar levels. So constant stress is associated with heart disease. And maybe this makes sense because if you're under constant cortisol release, you're going to see a much higher level of glucose circulating in the bloodstream, which means it's more likely to glycosylate the hemes and lead to plaque formation and hardening of the arteries and all that good stuff in there too. So the key is don't be stressed. Okay, easy to say, right? Yeah. Cortisol counteracts insulin. So insulin is like, get ready, here comes the sugar, get ready to store it. Cortisol is like, release the sugar, we need to use it. And again, continue to release the cortisol, you get that adrenal fatigue. So you kind of get, I want to say like an adaptation to cortisol in there. And if your cortisol starts not having too many effects, then maybe you see its release ultimately become compromised. And ultimately you can have issues with a damage to various tissues, including bone, the immune system, because you start to lack a response to the stress factors that are in there. So there's not a way, the body does not work to correct the deficiencies that would normally cause the cortisol release. So I know we talk sometimes in various health aspects of people who spiral down. This continued cortisol release would be an example that could lead to a spiraling down where you release cortisol because you have these horrible effects, but the effects keep staying there. So you release more cortisol, you eventually develop an insensitivity to cortisol. So you're not responding to the stressors that are in there. So there's no healing that's taking place. So you kind of get that lovely little spiral. So from a field. Yeah. And this is a tough a tougher type of thing to treat, I think, in some cases in there. So yeah. Yeah.
But it all started.
All right, so one of the ways that this chapter kind of wraps up is talking about a lot of these effects, these hormonal controls, using the example of the diabetes. I got to get my Wilford Brimley mustache and a bowl of oatmeal, and like he was a mainstay on television when I was a kid. So, but there's two types of diabetes. Actually, there are more. You also have gestational diabetes, and then there's another weird form of diabetes that can come up through some. But the primary ones we refer to as type 1 and type 2. Type 1, your body just doesn't make enough insulin. So usually this is due to some type of damage to the beta cells, often through an autoimmune disorder where your own body destroys the beta cells in there. This used to be called juvenile diabetes because it would occur most frequently in young people. It could also be called insulin dependent because those are the people who need to take injections of insulin regularly after their meals. Type 2 is now very common, usually later in adulthood, and this is an insulin resistance where your body is making the insulin, but your cells just aren't responding to it. Usually this becomes associated with obesity, and it just kind of spirals out of control. That's going to be our theme for the day. In both forms of diabetes, though, the circulating concentration of glucose in the bloodstream is elevated. The body tries to dilute it. So if you're constantly thirsty and constantly urinating, that is one of the early signs of maybe pre-diabetes because you're trying to dilute this sugar in the bloodstream. Constantly thirsty and constantly urinating. Okay. What's that?
I was like, that's me. I remember when my brother was younger, he had like a bad urinary problem. He was always thirsty.
Yeah.
My mom got tested for diabetes, but it's just.
Yep. And there are other things that can cause that as well too. I mean, if you are really circumspect, like I don't know if you see, I always think of like, the women at the gym who carry like the gallon bottle and are chugging down the one. Yeah. Yeah. Yeah.
I used to drink like one of these like hour and a half. At one point for like 3 years.
Yep.
My stepmom was like, my stepmom and dad started and they were like, you can go to the
Yeah. And I mean, if you flew it in, flew it out. So that's what the body's going to do there too. And drinking a lot of water is good for you. Little caveat, if you like chug a gallon or 2 gallons of water, you can drink enough water that you can get intoxicated. There is something called water intoxication. At that point, though, you are close to, you're close to a bit serious electrolyte imbalance, and you can die if you, yeah, everything kills you.
potassium levels.
Yeah. Yes. People ask, how you want to die, and I've got an answer for that. I want to die with the heat death of the universe. So there you go. I figure I got a few billion years at least. Yeah. Don't think that's how I'm going to die, but yeah. Me and Walt Disney. No, okay. But type 1 diabetes, what we see is that fat breakdown is accelerated. All right. So you're like, wait a minute, isn't diabetes associated with, obesity? That's primarily type 2 diabetes. Type 1, you often see these very skinny kids because they can't keep on body fat. And we see high production of ketone bodies. Some of these ketones are keto acids. So that can lead to ketoacidosis, where you have an elevated blood pH. And in this case, the bicarbonate buffering system, you can exceed its capacity to buffer all these keto acids. This is the people whose breath might smell like acetone. They're undergoing ketoacidosis in there. So untreated diabetes of this type will lead to a significant weight loss. All right. Dang it, stop it. All right. So what happens if we have elevated blood glucose levels? High fasting blood glucose is a warning sign for diabetes. Normally, your fasting range would be, I think, between 70 to 90 millimolar per deciliter or something like that. Don't quote me on those units in there. A low fasting blood glucose is a sign of hypoglycemia. So low blood sugar can lead to other issues as well too. I think there was a previous slide that kind of looked, if you get below 40, you're gonna get the shakes, and you start below 30, you start to get in the risk of going into a diabetic coma and all that good stuff in there. Normally though, after eating a meal, you can see your blood glucose levels spike up to 140, 150, and that's, milligrams per milliliter. Okay. So actually, I had to go back up to the starvation. Let me find the starvation one here. Okay, you can see that for starvation, your blood glucose is going to, your body's going to work to keep glucose circulating in there. So even after like a month of starvation, your blood glucose levels are going to be at about 30. Now that's low, that's bad, that's dangerous. It's when you get below 30. If you get to like 10, that's coma and death right there. But your body is going to exhaust everything it can to keep those glucose levels okay. And of course, your brain is going to be fed a steady diet of the ketone bodies. So as long as you have enough water, that's why you can survive for like a month without food, as long as you have water there. Okay, long-term effects. I mentioned this when we talk about glycosylation. If you have lots of glucose in the sugar, you got all those hydroxyl groups. Those are good for proteolytic attack. So you can glycosylate particularly free amino groups. Hemoglobin has lots of those exposed amino groups. So glucose can go in there and glycosylate them, and then that will compromise your oxygen delivery. One of the tests they do for diabetes, the extremities, they will kind of run the little pinwheel over them to test for sensation, because you can result in nerve damage. I believe that's through lack of oxygen being through there. And I have to tell a horrible story to you guys. So, my girlfriend in grad school had a brain tumor, so I spent a lot of time in the ER with her when stuff would happen. So I would hear other things. We were in a waiting room waiting to see. And but at this point, we were comfortable with the seizures and just getting them checked out. But near us, there was a woman who was kind of whining and crying about the pain in her legs and all that. And, it was basically late-term diabetes, and they were talking they were going to have to amputate her foot, and it sounded so horrible until we realized that the patient was a nurse in the diabetic ward and had just been non-compliant. It's like, you see this every day. And that what that does is that, kind of strikes the chord of sometimes it is incredibly difficult for patients to adhere. Yeah. Denial, or just, unable to, for some reason, unable to follow a regimen that would keep her healthy. And, it's one of these things where it's like, oh,
hours, it doesn't really allow them to take it.
yes, yeah, yep. And it's easy to, I'm gonna make fun of you, go grab a cheeseburger or something like that. I'm like, woo, I haven't had I've had like one cheeseburger in the past 40 days. And like, oh, okay, well, I don't know, I just saw the wrapper in there. Everything looks like a cheeseburger to me. Yeah. But I had 2 pieces of pizza, and then I was really bad one day, cutting down on sugar. But my daughter had this thing at school, and people brought a whole bunch of food, and so I've been trying to avoid like processed meats. But they had the little cocktail weenies wrapped in something, and I was like, oh, these are so good. And I was like, oh, look, a brownie, and I had so it wasn't like a full size can of Coke. It was like one of those little 8-ounce cans of Pepsi. I was like, oh yeah, but yeah, exactly. Like, that was like my one bad day, and woo, yeah. But it was all so delicious. But because of this hemoglobin becoming glycosylated, of course, then you can see the macrophages attacking those and forming the foam cells, and resulting in the plaque formation in there. And you get that damage to the small blood vessels and nerves. Again, primarily these are going to be in extremities. So that's why ever in doctor's offices say, diabetic patients, please remove your shoes and socks. It's because they're going to test sensation in the feet and the toes to test the level of damage to the. Yep. And then you have people who have like varicose veins or some type of circulation issue that have to wear the compression socks, and those are really, really helpful. But I'm like, how do those? Actually, I guess they're more helpful than anything else. Yeah. Adipose tissue releases something called adipokines. These are basically hormones that originate from the adipose tissue. So we tend to think about, and most of the way we lecture about adipose is like, it's where you store all the fat. Oh, hormone sensitive lipase, you cycle out free fatty acids and diacyl and triacylglycerides and glycerols in there and all that good stuff. It just releases fatty acids or brings them in. But it also functions as an endocrine organ because it does release multiple hormones in there. There are these adipokines that carry information about the fuel storage to the brain. These include leptin and adiponectin. Remind me, in case I forget, I want to talk about gastrin as well too. Gastrin, yeah, that's actually from the stomach, not the adipocytes, but yeah. So, what we got here? So leptin is this lovely little appetite suppressant. Okay, so if you're full, if you have enough energy, if your energy stores are full and you send some triacylglycerols to the adipocytes, that's going to release leptin, send that to the brain and say, hey, we're good. And leptin acts as an appetite suppressant, tells your brain to say, don't tell the rest of the body you don't need food. So it should suppress hunger. This adipokine was first identified in obese mice, and they called it as a product of the OB gene, probably for obesity in there. So homozygous OB mice ate continually. And because they ate continually, they had elevated cortisol, they shivered, they didn't reproduce, they had insulin resistance, and they were obese. And you'll see a picture of them. They're so cute and fluffy, but they're these big fat mice. When leptin was injected into these mice, they lost weight, their body temperature turned to normal, and they became much healthier. Okay, so the adipose tissue releases leptin, goes to the brain, says, stop eating. All right. So if we compare this, here we have our homozygous OB mouse on the left, and here we have a homozygous OB mouse also on the left, but who has been injected with leptin. All right. So clearly there was some issue either in the production of leptin or, well, it had to be production or transport of leptin. And here the leptin actually got to the brain and kind of corrected the error. So you can see our big squishy fuzzball of a mouse there. And here's our other one. I always think this one looks like it's got like tentacles growing out there because of the photograph of where its foot is underneath its chin. But as you can actually see its feet, whereas this mouse is so fat and fluffy that you can't even see its feet. So, all right. Anyway, there could also be errors in the leptin receptor as well. So maybe the adipose tissue was producing leptin, but it just wasn't binding to the receptor. And because it wasn't binding, it wasn't triggering the intercellular effects in the brain. In essence, the signal wasn't getting through. So these mice would also continually eat in there as well too. And you could see this receptor in the hypothalamus. And again, these mice were obese and diabetic. So leptin is associated with eating behavior. Now you can imagine when this was discovered, the pharmaceutical companies just salivating. It's like, we're going to be billionaires. We can use leptin to treat people as an appetite suppressant. And that's one of the things that does not show up in here. Actually, does it? Is it in here? It is. Yeah. Mm-hmm. Yep. Did it work? No, because leptin, clearly there are other factors in humans that are at play here, because numerous clinical trials looked at giving people leptin and no effect. Now, clearly it does play a role in there, but there are multiple things that affect appetite and eating and satiety and all sorts of stuff like that. We'll talk about gastrin here in a minute, and ghrelin. Ghrelin is the hunger thing. So, so anyway, it was a big disappointment because, it's like, we'll be billionaires. And, no. Okay, so how does leptin actually work? Once it binds, it actually affects a couple other appetite-related hormones, okay? So we're gonna look at a couple of these. A neuropeptide Y, awesome name, okay, not so much, but NPY for abbreviation is an orexigenic hormone. So orexigenic is appetite stimulating, anorexigenic is an appetite suppressant. So if you think of anorexia, I'm just not gonna eat anything. So that's what we see down here with our alpha melanocyte stimulating hormone. But neuropeptide Y sends the signal, go eat, go eat. If you starve yourself, if you don't eat, the levels of neuropeptide rise. So if you don't eat all day, by the end of the evening, you're going to feel really, really hungry, in part due to neuropeptide Y. What scientists found in those OB negative mice or DB mice, that the neuropeptide Y levels would increase significantly, but that they could be inhibited by leptin and by insulin. So leptin saying, I'm full, we've got enough energy. Insulin says, hey, there's fuel on the way. So both of those would have different pathways, but the same result, to decrease those levels. All right, so neuropeptide Y makes you want to eat. On the other hand, melanocyte stimulating hormone, or MSH, is an appetite suppressing hormone. So it sends a signal to stop eat. Its release is stimulated by leptin. All right, so you're like, wait a minute, okay, yeah, okay, that makes sense. Leptin, gets to the hypothalamus, stimulates the release of MSH, and kind of says, hey, we're full, we're all good here, stop, don't eat anymore. Okay, all right, and again, this is coming from our hypothalamus and our pituitary gland here. So our adipose tissue sends leptin to the hypothalamus. The hypothalamus sends the signals back saying, all right, we're full in here. We can see again, we have the adipose tissue, we got norepinephrine in here. Our G proteins, our cyclic AMP is a cyclic messenger. So we're going to increase expression of this UCP1, or uncoupling protein, which is going to signal our mitochondria to stop generating energy and just generate heat that way. We'd see this typically in our brown adipose tissue. So this might be something, our mice, the ones that were shivering all the time because they were constantly making energy, they weren't uncoupling that. When you gave them leptin, they go back to producing brown adipose, and uncouple that uncoupling protein would allow them to produce heat from the electron transport chain instead of producing ATP. Right? And we'd also see, stimulating the release of fatty acids in there to go into the mitochondria for beta oxidation as well. Okay. All right. So it may have something to do. The reason leptin doesn't work on humans, this is speculation. It might have something to do with the fact that rodents tend to have a higher proportion of brown adipose. So, like if we stop eating, we generally don't get cold. And if you do eat a meal, you generally don't get warm. If you drink a bunch of soup, something that's actually hot, then yeah, that can affect your body temperature. But we don't tend to have as much brown adipose. Perhaps if you spend a lot of time in the cold, maybe leptin would have a greater effect on you, but that would be, an interesting experimental design. You could actually kind of test that. But anyway, that's getting beyond here. But that's, the type of thing I'd want you to think about. Why is this working on rodents but not in humans? We see a lot of things that work on mice, but they don't translate well into humans, because while mice are similar to humans, they're not quite. Okay. So specifically, how does leptin work? Okay, so if we're going to look at this melanocyte stimulating hormone, leptin is going to bind to a receptor, and its receptor dimerizes when leptin binds. Okay, we then have this Janus kinase, and I think this is one of the questions I took out of an earlier exam, but now this is where it's been moved into this chapter. So this Janus kinase is JAK. It will phosphorylate a couple tyrosines in the receptor dimer, which that phosphorylation event allows these STAT proteins, signal transducers and activators of transcription, we'll just call them STAT, make stuff STAT, like your doctor shows, something like that. So the STATs will bind to that phosphorylated dimer, and then they become phosphorylated by the kinase, that Janus kinase. So we call this the JAK-STAT method. JAK-STAT method, not method, process, something, pathway, something like that. Activation cascade in there. But the STATs, once they become phosphorylated, they too dimerize, and that dimerization kicks them off of the scaffold in there, and they go to the nucleus and they stimulate transcription of the gene that controls MSH. Okay, so leptin increases transcription of the gene for the precursor to the anorexigenic alpha MSH. Okay, so we don't actually make MSH, we make a pre-pro protein, and then it gets modified to actually make the. So if we look at this pictorially, here's our leptin receptors, kind of separate this way. Leptin comes in, they bind and dimerize. When they dimerize, the JAK binds them and phosphorylates them. The phosphorylation is like the landing lights at the airport. The stat molecules then bind to those phosphorylated sections. Here comes the helicopter landing in. Once those are bound, Jack phosphorylates those. Then the stats dimerize, and then from that they go to the nucleus, and they start activating gene transcription. Okay. So I really, even though I don't emphasize this particular mechanism in here, I really like this picture because this, I think, demonstrates what's going on in good detail here. We have, our receptors, which are just kind of hanging out. When our substrate gets there, they dimerize. That allows the dimerization allows the kinase to phosphorylate them. Once they're phosphorylating, the way my brain works, I think, oh, landing lights. The stat comes in and binds to that. Well, they're now right beside the Jack kinase, so they get phosphorylated. They dimerize, go to the nucleus, have their transcription effects, and we can see all this nice little stuff going on. All right. If we think about this, you ever been to like this little thing that was a daddy-daughter dance, whatever stuff. It's like, oh, it's horrible. But my daughter wanted to go, so I go there. They had food. I grazed the whole time I was there. It's like, ooh, these little cocktail wieners wrapped in bread. Yeah, a little bit, but I was gonna get all crazy and do stuff in there, but like no. If exactly, I was totally gonna do the worm, or when they were playing some weird music, they did have a good variety of music in there, but it's like, oh, I think you need to play some Nucleus and let's jam on it, and go back and do some, throw down some cardboard, and I don't know if I can do those moves anymore. I was never a good break dancer, but I was better than average people. So yeah. But maybe if I'd known one or two people there, but whatever. Anywho, so grazing on all that food, it would take a while for this to happen. This is because it's a transcriptional event. We're talking, minutes to hours. So, you may eat a big meal, it may take like an hour to eat a meal, but then you're going to start to feel full in part because this stuff is happening. So it's not immediate short term like epinephrine, like, boom, bam, I'm full. It's like, oh, okay, now I'm feeling full as we start this transcriptional event. And then of course, as you get hungry, you stop producing leptin, all this stuff kind of, adaptation over time, degrades, and you stop producing these molecules and start the other way. So you kind of have this constant balance. It's like the seesaw of hunger fed. All right. Okay. So I sort of mentioned this earlier. Leptin will increase thermogenesis. So this is where that whole uncoupling protein, we saw that picture back here with the uncoupling protein. Neurons in the hypothalamus will send signals back to the adipose tissue and release norepinephrine. And then you get a whole bunch of these adrenergic receptors, big cascade of that. I think there's all cyclic AMPs and various kinases in there, but it ultimately results in a nuclear signal that will increase the transcription of this uncoupling protein. And so the fatty acids that get released in the adipocyte, the uncoupling protein, so you're oxidizing those, but instead of making ATP, you're just making heat. And so as you make that heat, you kind of shiver the fat away. I don't know, maybe this, I always think of those like 50s things where they had like the big strap on the machine that would, yeah. It's like, works so well. No, you find like an electric stimulator in there, just look, I got abs. I just had to shock myself, just had to tase myself. Yeah. And a lot of those infomercials are based in dubious science. There's an underlying kernel of truth, but maybe the application is not exactly rigidly well thought out. All right. Okay. So we know about leptin release in there, and for our OB mouse that doesn't make the leptin, we also have the leptin receptor. So our DBD mouse is not going to have those. So if you restore leptin to the mice that don't make it, they go back to the normal body mass. They go back to normal in here. But leptin administration to most obese people does not restore normal body mass. So here's the understatement of the entire semester. This was a great disappointment to the pharmaceutical industry. Literally a billions and billions of dollars, probably trillions of dollars had this worked. Just like, oh, damn it. Many middle managers died that day inside. And the funny thing is, leptin is actually elevated in many obese patients. Not most, not all, but many of them. So what that means is that something downstream of the leptin is not working right. So maybe there's some form of leptin resistance. Could be the leptin receptor. Hey, we could CRISPR that in, right? Oh, ooh. We got to go to China and convince some rural farmers of something. Yeah, I can. All right, sorry, we got him referring to Dr. Hay. Yeah. So that's it. And it's like, forget this job. I'm gonna be a trillionaire. I'll buy Putin's yacht and replace his gold with platinum. Yeah, they've been showing pictures of his $700 million yacht that has gold-plated toilet paper rolls. I mean, I mean, it's funny because you can think that, horrible things about Trump and all the terrible ways that he enriched himself, but he did that with shady real estate deals for the most part. Unlike Putin, who has a $700 million yacht with gold coated, it should be polonium coated. Oh, Dr. You were getting mad here. Yeah, that would be poetic justice. Anyway, you can see where my sympathies lie. Okay. But orexigenic neurons have insulin receptors as well too, which is not too unusual since the brain needs glucose in there. So when insulin binds to these receptors in the brain, it's going to stimulate the release of NPY, right? Or sorry, inhibit the release of NPY and stimulate MSH. Okay, so NPY, again, remember, is must eat. So if insulin binds, it says, no, we're good. We got food coming in, And if MSH is going to be like, hey, we're full, right? So that's our anorexigenic. What this also indicates is that there is a likelihood of crosstalk between the insulin and leptin pathways. So leptin binds in the brain, insulin binds in the brain, and they kind of have cross purposes in there. So leptin makes liver and muscle more sensitive to insulin. Okay, they have a common secondary messenger they could have a common secondary messenger that might enable both of them to trigger the same downstream pathways. All right. So if you're hungry and you start to eat, the leptin may be priming the rest of the body to respond more rapidly or more significantly to insulin. Okay. All right. So let's talk about a couple other hormones that control eating as well too. Okay, we've sort of mentioned our alpha MSH, our melanocyte stimulating, says eat less, metabolize more, we're full. Our neuropeptide Y says eat more, metabolize less, we're hungry. Okay, so you can see here we're making more metabolism, less cheeseburger, more cheeseburger. Should put a cat in there. Has cheeseburger? I'll name my cat neuropeptide Y. Oh, that's awesome. Okay, that's a good idea. Yeah, it's always something new every year I teach this class. Okay. Anyway, if we go back into the organs, we get to this stuff being released by the brain, by the hypothalamus. But we've talked about leptin and insulin, increasing MSH, decreasing neuropeptide Y, and these all come from the adipose. Okay. Our stomach, I think I mentioned ghrelin. If your stomach is growling, you're releasing ghrelin. Always makes me think of some event that was held at Gray Lynn where they had a big smorgasbord of food. I was like, oh, Gray Lynn, that was good food, because I was hungry and I don't know, whatever keeps the stuff straight in your minds, that's good. But ghrelin is also going to promote it will come from the stomach, go to the brain and say, hey, we're hungry, we need food, and induce the release of neuropeptide Y, which will result in a signal cascade of making you want to eat more. Yes. And that actually may be a physiological response. It's like if I get hungry, I don't usually think about my stomach getting warm or anything like that. But that just could be an effect of the release of some of these compounds in there. Your gut also. So if we look at our intestines, yeah. We also have a couple other compounds in there. There's the awesome peptide called PYY3-36. That's why we don't need to memorize them. You can tell different groups come up with different ways to name things here. And there's also this GLP-1. What does that stand for? That is our well, maybe it's not in here. Come on. Well, I forget what it stands for right now, but both PYY3-36 and GLP-1 come directly from the intestine, and as food flows in, that stimulates the release of these hormones, which signals the brain, hey, we've got food coming in, we don't need to eat as much. Okay. And of course, there are ways Coney Island hot dog eating thing, whatever something. You got to overcome this. No, I'm not full. No, I mean, this is probably not healthy to be able to eat 108 hot dogs in 10 minutes or something like that. But you can train your stomach to do stuff like that. I know when I've mentioned my gluttony training for going to the all-you-could-eat seafood bar and just eating plate upon plate upon plate of crab legs. The way I would do that is have like a cracker every hour, so that there's enough so that my stomach wouldn't contract, but I'd still stay hungry. So all this in there, so I could have a big, because if you don't eat for a long time, your stomach contracts. I wonder if maybe that's part of the warmness of it as well too. As your stomach contracts, maybe that's releasing heat or freeing up something in there. Yeah, it's all. Yeah, and then there may be like shakiness and all sorts of So it's just kind of recognizing these signals. And one of the things I like as an analogy for this is you see the commercials for Snickers bar where they take like someone who turns into gets really it's like, you're hungry, eat a Snickers. I used to when my wife was younger, sometimes she'd get mad and I'd say, you're tired, take a nap. And it was really true. It took me a while to recognize that, but and then she would recognize it as well too after a while. But yes, exactly. So you get cranky if you don't have enough food or enough sleep. All right. So before I jump into this, I did want to mention gastrin as well too. This is another gut-derived hormone. So I found a fun article that talked about like why do people poop after they drink coffee? Why does coffee make you poop? So they actually, there are theories. This is not something that's been studied in great detail, but there are a few studies that came out. And I just saw something published not too long ago. But good headline. And they did a control where they used warm water. They gave people warm water, and that stimulates some people to poop. But they gave people food, and that had along with water, and that had a greater response. And then coffee had the greatest response in there. So it may be the tannins or whatever the compounds are that are in coffee. Yeah. So I believe caffeine plays a role, because I think they actually tested caffeinated versus decaffeinated coffee. Decaffeinated coffee still was higher than warm water by itself. And caffeinated coffee, I think, was the highest. I'll have to find that article in there. Yeah. And right, so and it depends. Again, with dairy, many people have some level of lactose intolerance. So
they said we are the only species that is other animals milk. They say we're really not supposed to drink, cow's milk.
Right. Yep.
But
And
I do learn that everybody has to go to the back, so some people are
yep. There was some mutation that occurred, I want to say somewhere in the Caucasus Mountains, like thousands of years ago, that allowed people to consume that without an issue. Of course, I look at cats that drink milk as well too. I'm like, okay. So I don't think we're the only ones, but we're the only ones that get a primary source of sustenance from other animals' milk. Yeah. Yeah. And it can. And if you look at, I think the Asian population has the high, it might be, it's like 75-80%. So one of the things that I noticed, going to Asia is you don't see a lot of cheese. Yeah. But the theory between the caffeine and some of those other small chemicals that come from the coffee bean, they may be stimulating the production of gastrin, and gastrin is associated perhaps with inducing bowel movements. All right, so, and again, it's another hormone that comes from our guts. So we tend to think hormones, hypothalamus, adrenal glands, or maybe the gonads or something like that. But here we have our fat cells, our stomach, our intestines. They all produce hormones. So we have this constant interplay between the different organs that are helping to regulate the entire organism. And that is our key focus of the entire chapter there. Okay. Another hormone that we see is something called adiponectin. And again, this comes from the adipose tissue. Conveniently, most of the adipose-derived hormones start with adipo in there. But adiponectin gets released by the adipose tissue, circulates, and again, enhances other organ sensitivity to insulin. Okay, so this is not well understood, but it is an AMP activated kinase pathway, which is different from a cyclic AMP activated kinase pathway in here. But one of the neat things that happens here is we're going to inactivate our ACC, our acetyl-CoA carboxylase, which would make malonyl-CoA, which would inhibit fatty acids import into mitochondria. So, we may be enhancing the cells to use the glucose that's coming from the insulin that's in there. Okay. The other thing is AMPK also inhibits cholesterol synthesis. So we're saying, hey, use this glucose. Okay. If we look at this, our AMP kinase for regulating ATP metabolism is interesting. This looks like a terrible picture here. Okay, but here's our AMP kinase. When AMP kinase is active, it will inhibit ATP consuming processes such as biosynthesis. We're not going to make our cholesterol. We're not going to make fatty acids. It's going to inhibit transport, inhibit muscle contraction, inhibit growth promotion, all sorts of stuff like that, things that would consume ATP. Okay, it will promote ATP producing processes like oxidizing glucose. So you can see, well, hey, if we take in a meal, we have a lot of glucose circulating in the bloodstream. Insulin, so AMP interacting with insulin is going to make that more sensitive so that we can oxidize glucose and oxidize our fatty acids in there. On the other hand, what we see is ATP. When ATP levels rise, it's going to inhibit our AMP kinase because we don't need to make as much ATP, and we can spend ATP on these ATP consuming processes. Okay. We can also look at levels of ADP. Okay, so ADP may get used here. This is kind of seems like a terrible feudal cycle type deal. ATP plus AMP can give you 2 ADPs, but our ADPs can then result in increased level of AMP, which can elevate our AMP kinase. So this is kind of like a feed forward in here. Things that we see that happen that alter this pathway: hypoxia. So if we lack oxygen in there, or if we have glucose starvation, we go on a sugar-free stuff like that, or some types of metabolic poisons will actually kind of short circuit this this process and disrupt this greatly. Okay. If we look at adiponectin, so adiponectin has a lot of effects across a lot of organs in here. So again, if we have a long fast or starvation, we're going to see adiponectin released from the adipose tissue, and we're going to see this elevation in AMP kinase. Same thing can happen through exercise as well too. So exercise, how do you lose weight? Starvation or exercise? It's the same thing. All right. But what we'll see is an elevation in cardiac glycolysis through the action of the PFK1 in there. So here's our target enzyme. Yeah, our GLUT1 and GLUT4 transporters get elevated, so we're going to bring in more glucose into these tissues in here. We're going to increase fatty acid oxidation and decrease fatty acid synthesis. We're using energy, that makes sense. Lipolysis, we're going to decrease lipolysis, so we're going to see an inhibition of our hormone-sensitive lipase. We're not going to be making as many triacylglycerides. And it's funny, this is one of those things where I caution you, just because we got that big red X there doesn't mean we stop it. We're just slowing it down, because 75% of the free fatty acids that are released within the adipose tissue are immediately reesterified back into a triacylglycerol. No matter how long you go through the starvation, you just might have fewer total triacylglycerides in there, but that 75% recycling, immediate recycling, just seems to be pretty constant. And you can see these other effects in here as well too, on glycogen synthesis, protein synthesis, and all that good stuff in there. Okay. We can look at the target tissues, what happens in the heart. We increase fatty acid oxidation, increase glucose uptake and glycolysis. Skeletal muscle, just no glycolysis in there. The brain, we increase feeding behavior, we decrease energy expenditure. So if you get hungry, you sometimes get tired. I can't think straight, I need a Snickers bar, right? Today ought to be sponsored by Snickers. Okay. Again, if we have adiponectin, that means we don't have lots of circulating glucose, so we're going to see a decrease in insulin secretion from the pancreas. Our liver is going to stop synthesizing fatty acids and cholesterol, and our adipose is going to decrease fatty acid synthesis and that whole lipolysis in there. All right. Okay. Our PPARs, okay, proliferator activated receptors, alter the expression of genes for fat and carbohydrate metabolism. So if you change your diet, you're going to see a big change in this. So they're named because they were discovered in peroxisomes. They typically bind fatty acids and derivatives. And we had mentioned this retinoid X receptor before. All cells have at least one form of retinoid X receptor. But these PPARs can bind to the retinoid X receptor and have significant transcriptional effects in there. So of course there are several types: alpha, gamma, delta. It's like, where's beta and why are they not in the right order? That's but the functions of them, PPAR gamma is in the liver and adipose tissue. It turns on genes for lipid synthesis and storage. It can be activated by a variety of compounds such as thiazolidinediones. All right. Our alpha form is in the liver, heart, and the skeletal muscle. Pretty much any place that's going to be using them. It's activated by fatty acids and eicosanoids, and it turns on genes to uptake and oxidize those fatty acids. Okay. And delta is in liver and muscle, and turns on genes for fatty acid oxidation and mitochondrial uncouplingly. So again, this would be our producing heat to some extent. How do they work? So why do I talk about PPARs? Because of their mode of action. And again, this is just illustrative of how things can work. So what we see is our ligand comes in here, gets into our cell and binds to the PPAR, all right, which is going to have another ligand bind to the retinoid X receptor. Once those two both have ligands bound, they dimerize, so we make a heterodimer, and then that heterodimer goes to the nucleus, binds to the DNA, and it activates transcription. Okay, or regulates transcription in there. So the key is ligand binds to PPAR, different ligand binds to the retinoid X receptor. The two together with those on dimerize and go to the nucleus and activate transcription. All changes. All right, so PPAR delta, key regulator of fat metabolism. Mice who are overfed do not become obese if PPAR delta is constitutively active. Up until a couple of years ago, I used to joke, I guess my PPAR delta is constitutively active because, when I was younger, I would drink a 2-liter Mountain Dew and eat like a big bag of Doritos pretty much every day back when I had scholarship money, and it's like, woohoo, no, not gain weight, just stay skinny. Lunch ladies would give me extra portions because they felt bad for me. That's how skinny I was constantly. If we look at those mice who lack the leptin receptor, have a leptin receptor deficiency, the PPAR delta overcomes that and prevents obesity in those mice. Its major target seems to be mitochondrial uncoupling. So we oxidize the fatty acids and burn them and produce heat, and we don't produce energy. So we can keep burning that fatty acid in there. As long as we're doing that uncoupling, we can just kind of burn that through there. Okay. This is just a overview of our different PPARs in there. So our PPAR delta, the big thing is thermogenesis in the adipose tissue. So we're promoting brown adipose as opposed to white adipose, which is just storing it. And we even can get thermogenesis within the muscles as well too. It's like, ooh, interesting. Okay. So you can bet that that's being studied for that. We start to get into more stuff in here. mTORC. What does mTORC stand for? Let me see if I can. I hope it's down here. Mitochondria. Where is it? All right. Well, who knows? Didn't put that one in there. Yeah. But it's this is actually important for development and growth. So this is why we want to work to improve food security for the surrounding community, because when you are adequately fed, if you have good nutrition, then you can grow up big and strong. Again, this is reasons why a lot of pediatricians will evaluate kids on their ability to thrive, and failure to thrive becomes an issue. Are they growing? Are they getting bigger? Are they being active? And so on. So, even kids who are obnoxious and loud and whatnot, that's what kids are supposed to do, because they're growing and they're getting bigger and they're thriving. Okay. It's the ones that are kind of weak or don't do anything or they have poor muscle tone that you worry about. So mTORC is involved in this process of growth and development. So you can see it plays roles in glucose uptake. It affects ribosome and protein synthesis, mitochondrial metabolism, angiogenesis, adipogenesis as well too. So all sorts of stuff can kind of be attributed to this mTORC signaling molecule, which is activated through the presence of nutrients and various growth factors. I don't know if in biology you may have talked about like VEGF and EGF and all sorts of hormone, there might be some, but all these growth factors. So even from early development, from early egg, you can see the release of these, and then kind of the various concentrations in the developing embryo, whether you get three types, mesoderm, ectoderm, or endoderm. Is that it? Yeah, I think, but it kind of depends on the gradient in there. So if you have a high concentration, you get one type of tissue. Your neural crest tube and the way all this stuff folds in here comes from a lot of these growth factors. All right. Our different class of drugs here activate the AMP and PPAR pathways, and they can increase the expression of adiptonectin genes. A lot of these are used to treat type 2 diabetes. So if you look at Avandia, that is a very commonly prescribed diabetes drug, and Actos. So these include receptors that lead to the activation of the adiponectin gene transcription pathway, the AMPK pathway, and our PPAR gamma. All right, so we can try and induce thermogenesis in here. Avandia is limited in its use because it does increase the risk of heart disease. Like, oh crap, man. So advantages, drawbacks in here. You ever listen to like the commercials for different drugs, and they go through the list of the side effects, and you're like, yeah, I'd rather have the illness. This is one of those things where you balance it out. If you have very severe uncontrolled diabetes, then the risks are probably worth it. And as we get better at genotyping people, we can start developing genotypic specific regimens that are optimal for each phenotype. All right. Oh, ghrelin, here we go. Short-term or orexigenic drug in there. Ghrelin receptors in the brain, the heart, and the adipose tissue. Again, not well understood, but it's a G protein coupled, easy to remember, ghrelin, G protein coupled receptor, and increases the sensation of hunger. So it's kind of neat. You can inject people with ghrelin and they'll get hungry. Where is this useful? Cancer patients, because of the lining of the stomach, fast growing cells, those are often a collateral damage of cancer drugs that target fast growing cells. So you can have this kind of stomach upset. You don't feel like eating because you've killed off all these cells on the lining of your stomach. Checked with ghrelin, stimulates appetite. And some of you are probably sitting there thinking, you can also tell them to smoke some weed too, right? Because that stimulates appetite as well. So,
Really?
yeah. It's just unexpected. Yeah, but that's in there. If we look at Prader-Willi syndrome, yeah, and Prader-Willi, I think, I don't know if you recall, you end up chewing off the fingertips and stuff in here. It's a horrible neurodegenerative disease in here. If ghrelin is constitutively active, you're actually, no, it's not the chewing off the fingertips, that's a different one. But Prader-Willi is a constant eating. I think these are the people who like eat their own hair and whatnot. It's just insatiable appetite. And they tend to be, huge and obese because they're always hungry, because, the ghrelin is always active in there. So finding ways to counteract ghrelin would become necessary for Prader-Willi disease treatment. If we look at relative levels, I like this picture because it just kind of gives you an idea of what's happening here. You have your breakfast, okay. So you wake up, and your ghrelin levels are elevated. You get hungry, you eat breakfast. Ghrelin levels drop off almost immediately in here. As you eat breakfast, your glucose levels rise, shown in the blue here, and of course your insulin rises concomitantly with that as well too. All right. Over time, your glucose levels will drop, your insulin levels will drop. You will get more hungry. As it approaches lunchtime, you can see right before lunch, your ghrelin levels start rising, right? And you say, oh, okay, now I'm hungry. So you eat something at lunchtime and you see, boom, glucose and insulin levels rise, ghrelin drops off. And this repeats for dinner and maybe a snack and so on. And then over at nighttime, your ghrelin levels rise. I think this should probably wrap around and be at the same level. That bothers me. Like these are pretty close together, but why is ghrelin so high and then it's lower over here when you wake up? No, okay, that's just the chart or the data that they got for one person. Apparently this person was hungry all night. Gonna wake up in the middle of the night and eat something. All right, PYY 336 is an appetite suppressant. Again, this comes from the gut, small intestine, and the colon. So it's named because it's got 36 amino acids and 2 tyrosine residues at the end. So that's where the YY comes from. But it's secreted in response to food entering the stomach. So once you get food in the stomach, you release PYY 336, goes to the hypothalamus and says, hey, we got food. We don't need any more. So you ever like overeat? I remember 2 friends of mine went to pick up food. There were 7 of us that were hanging out at my house, and 2 of my friends, and they were bigger guys, but they went to Long John Silver's and they asked for like 10 pieces of chicken and 20 pieces of fish or something like that, And just yeah. It's actually, it's yeah. Long John Silver's has a special place in my heart because that was like the fancy place to go to eat when I was a kid. Yeah, we were poor growing up apparently, but they asked for like 30 or 40 pieces of food there between the 2 of them, and the guy at the counter is like, is that for here to go? And that's when they're like, yeah, I think we might be fat. So but if you start eating food, it's oftentimes hard to overeat, because you get this release of PYY 336, and if you continue to eat, that's when you get that stuffed feeling like, oh, I overate, oh, too much food, or whatnot there. So when you start to eat and start getting food in there to satisfy the energy needs of your body, that's when you'll see this release of PYY 336. It'll reduce the hunger. There's also another aspect to this as well too. We talk about the hormones that are released from us as organisms, but we have some symbiotic organisms within us, our gut bacteria. And I don't know if you guys have heard about like the poop transplants. And if you are healthy, there are places that will actually like buy your poop. That sounds terrible, and convert it into a pill form to give to people to improve their gut bacteria as well too. So yeah, renewable resource. Okay. But it turns out if you look at the types of bacteria in the guts of different people, they vary. Obese and lean people have different gut bacteria. Some microorganisms create fermentation products that will affect the adipose tissue as well too. Many of these are short chain fatty acids. Okay, so a lot of these short chain fatty acids freely diffuse across there, and these can have effect like our G protein coupled receptors of precursor cells, inducing them to become adipocytes and inhibiting lipolysis. So if you have a lot of propionate, if you have a lot of gut organisms that make propionate, you're more likely to make more adipocytes, store more fat, and not burn as much fat in there, right? In our lovely picture in here, yeah, so gut microbiota produce all these short chain fatty acids, which can affect energy availability and these protein signaling in here. All right. How much more do we have? Oh, good. Good. We're not too far ahead here. Oh, shoot. Yeah, but we're running low on time. So let's fly through these last 4 slides. Metabolic syndrome, this is believed to affect about a quarter of the adult population. We may be up to about a third that are diabetic or pre-diabetic, and the hallmark is insulin resistance in here. There are a cluster of symptoms. Abdominal obesity, tend to be fat in the middle, high triglycerides, low HDL, high blood pressure, elevated blood glucose, and various signs of inflammation. It could be like a neuropathy, extremal neuropathy, and fun stuff like that. So how do we get, on the other hand, there are many people who we would think are obese, but who are very, very healthy, healthy fat versus, fat skinny and, all sorts of stuff like that too. So 80% of type 2 diabetics are obese, but that's still only a minority of obese individuals who develop diabetes, because something like 40% of the population is obese. Probably more than that now. Adipocytes become packed and they're unable to accommodate more triglycerols. So what that means is that fatty acids accumulate in the bloodstream, and those fatty acids enter the muscle and liver. You get fatty liver, you get triglyceride lipid droplets, and that induces a loss of sensitivity to insulin, which ultimately means blood glucose levels rise. We see glycosylation, and heart disease and all that fun stuff in there. So the key is we don't want to overload our adipocytes. So we have these nice little small adipocytes. And if you get overweight, you can get larger adipocytes. And it's not like we can change the number of adipocytes. We can just change kind of their size in there. So we have this pro-inflammatory state where we have these macrophages in there. There are 2 types of macrophages that are significant here. Type 1 is more like the, oh no, let's just fix something, throw something up there. Type 2 is a let's repair things. So elevated macrophage 1 is usually going to result in scarring, or those are the ones that are going to lead to foam cells and plaque formation and arthrosclerosis and all that good stuff in here. And you can also see the macrophages infiltrate the adipose tissue in response to that MCP1, and you result in plaque formation there. Export fatty acids. Fatty acids go to muscle where they get deposits in there. If you think about steaks, look at the marbling in this, and like a lot of people like prime rib because of the fat that runs through there. And I'm like, no, I like a really lean cut of meat. So our cows are well fed. If they're too well fed, you get adipose in the muscle, and that's where that marbling comes from there. But anyway, ectopic lipids then interfere with the GLUT4, which means that it can't uptake glucose as well. So you end up with a higher level of circulating glucose. And we've talked about the problems that leads to. So how do we manage type 2 diabetes? Diet and exercise. It's a huge thing in there too. Control the diet. Don't eat a lot of fatty foods, or high levels of sugar. And it's funny because I think of diet drinks. There are some artificial sugars that have, an even greater glycemic impact than sugar by itself. Yeah. So, and that's something that was only recently come to light. A lot of these glycemic impacts of diet drinks can have a greater health effect than if you just drank sugar in there, pure sugar water. Yeah. Yep. Yeah. Yep. So, processed foods, a lot of, all sorts of stuff. So insulin, some people may need to supplement with insulin. Metformin, which is going to activate the glucose in there today. So you perk up with that. So many people. Yeah. Mm-hmm. Yep. PPAR activators as well, these different thiazide drugs, stimulation of insulin by binding to sulfonylureas can do that. So some sulfur drugs. A lot of people don't tolerate sulfur drugs, but they can work to improve this as well too. and preventing proteolytic degradation of glucagon-like peptide. That's it. GLP is glucagon-like peptide 1, which stimulates the insulin stuff there. So this is where things like Januvia come into play. All right. And I would suggest maybe even CRISPR therapy. Yeah, but it's unethical. So anyway, CRISPR. So going and genetically fix things in there. So maybe that can affect, like if you have damaged beta cells from an autoimmune attack, maybe you can repair the pancreatic beta cells to release stuff. Or if you have, poor receptors, but that requires a lot more information and is expensive and speculative at this point. Anyway, to summarize the entire semester so far, everything that lines up here, our nervous system controls the production of specific hormones. Other tissues also produce hormones that go to the nervous system and affect these as well too. So we can have a variety of types of hormones, but they can act throughout the body. We want to keep in mind that some hormones act in autocrine system. The cell releases hormones that immediately bind to receptors on the surface of that cell and affect it. Paracrine, nearby, endocrine, long distance in there. Be familiar with diabetes, insulin, what it does and all that good stuff in there, and all sorts of stuff in there. All sorts of stuff. Okay. So the other thing I want to take away from this is I'm going to give you guys your next exam on Tuesday. Like, what? That's terrible. Mean Dr. Ebert. Let me find the review in here. It is going to be a completely take-home exam. So that's going to be. Yeah, yes, yeah. You can use all your non-living resources. Yeah. So yeah. But where is that? Exam 3 study guide. I want to check on this just to make sure, because I haven't had a chance to look at this one. But like I said, class on Tuesday so that you will receive your exam, and then I'll have a Dropbox. Yeah. Ideally, you'll bring it back to me on Thursday, but I'll have a Dropbox as well too for you to submit it, I think. Come on. Stop. Yes.
I didn't wait till the last minute to get that until I was 12 hours.
Yeah. Yeah. Not the we can go ahead and download it in case I have comments. Yeah, good. All right. So for Tuesday, be familiar with fatty acid synthesis, know about triglycerols, what they're made of, where they're found, how they are made and how they are metabolized, and know about cholesterol as well too. Okay? Being able to distinguish between these, you're definitely going to see a question on cholesterol. In the past, I used to ask a question. There was a news article that talked about good cholesterol versus bad cholesterol, but now that's really far out of date. And so I would just ask you to analyze that article, but you may find something like that. Amino acid biosynthesis, you want to look at your precursors in there. Why is glutamate, glutamine important? Shorthand version, temporary repository for nitrogen, and also they often feed into so many other of the metabolic pathways in there. How do we make nucleotides? How do we degrade them? And then we get into our hormonal regulation here as well too. How does the body kind of maintain mass over time? What happens when you eat a meal? What happens if you don't eat a meal? What happens if you change your diet from all carbohydrates to all lipids? That might be a good question. I gotta get rid of all my questions. Sometimes I like my questions too much, but just kind of take a look at this, and this will give you the ideas that I'll focus on. I have most of my exam made. It's now just figuring out which questions I actually want to put on there, and that is what we will do next Tuesday. But thank you for giving me an extra 6 minutes here. I will stop now, and yeah, the crush is coming.